Okay, let's continue to look at dihybrid inheritance. Dihybrid inheritance involves inheritance of two characteristics, where each characteristic is controlled by a different gene located at a different locus. Okay, let's take a look of this picture of a pair of homologous chromosomes. Just want to show you that these two are alleles, represent a gene, and they are, for example, A here and C, they are located at different locus. So this is the meaning of a different gene located at a different locus. Dihybrid inheritance involves two characteristics. So let's look at this figure 11.7. This is the schematic diagram of dihybrid inheritance for these two characteristics, the seed color and the seed shape. Okay, these two characteristics. And we have the two purebred parents right here. Each have their pair of homozygous dominant, characterized by all capital letter alleles, and another pair of homozygous recessive alleles, characterized by all small letter. So fertilization between these two purebred parents produce offsprings in F1 generation with all dominant traits of round and yellow seeds, all. Okay, so how to draw a schematic diagram? Let's revise, which we have already discussed earlier on when we did monohybrid inheritance. So first, we have to write down the parental phenotype, and that is round and yellow seed, constricted and green seed. Okay, this is what we see with our eyes. This is phenotype. Okay, next, from the phenotype, the parental phenotype, we have to write down the parental genotype, characterized by letters, capital letter, small letter. Okay, and then number 3 is that there is meiosis process to take place. So remember to write, to put an arrow here. And meiosis produces gamete. Number 4, you write down gamete. So there is an allele that is inherited from a pair of allele. Okay, so from here, seed shape characteristic is controlled by a pair of B alleles, whereas seed color, it is controlled by K alleles. Okay, next one is fertilization. Fertilization between the gametes produces F1, first filial generation. First, we write down the genotype. Okay, so this is the letter. This is the combination of the F1 genotype. That means from here we can depict their phenotype. That means what can be observed. Okay, so only round yellow seeds are observed in the first filial generation. Okay, next is we proceed to self-cross the F1 generation. So now the F1 generation, they are the parental phenotype in this case. Okay, mating happen. So we proceed. This is like the parental phenotype, round and yellow seed, round and yellow seed. Okay, next is we write down the genotype characterized by letter. This is heterozygous, heterozygous. The third word that we have to write, it's the same as the first one just now. We just repeat it. Meiosis happens, so we put arrow to form gamete. So these are the possibilities of gamete that are formed. So during formation of gametes, any allele for seed shape can pair with any allele for seed color. So that's why you have this combination. Okay, move on. When the F1 generation produced from dihybrid cross from the figure just now, okay, this figure was self-crossed, we have 16 of the F2 genotype combinations produced. 16. And the cross shows 4 phenotypic characteristics. 4 phenotypic characteristics here: round yellow seed, round green seed, constricted yellow seed, and also constricted green seed. So the outcome is presented here in the Punnett square. So this from here, this is horizontally, we arrange, we place all the male gametes, and vertically down, we place all the female gamete here. So we transfer from this, okay, respectively, and then this is how we work it on. B, K, B, K. So we place it here in the first box. Okay, subsequently, we follow. Okay, this is how we end up getting all the possibilities of the genotype. So from here, we came up with this phenotypic ratio produced in F2 generation. There'll be 9, total of 9 yellow seed, round yellow seed. So here 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, the second probability is that round green seed, 3 of them. 1, 2, 3. These are all ratios, yeah, probability. And the third probability is constricted yellow seed. This we can see here 1, 2, and 3. And we have probability, the smallest probability of having constricted green seed, and that is homozygous recessive. Okay, now therefore from this dihybrid cross, Mendel introduced Mendel's second law, also known as the law of independent assortment, which states during gamete formation, each allele from a pair of alleles can combine randomly with any allele from another pair of allele. Now this is the summary of dihybrid, Mendel dihybrid experiment. The new combinations of characteristics produced in F2 generation, namely yellow seed and round green seed, yellow constricted and round green seed, okay, and then 2 characteristics, seed shape and also the seed color, are combined in the first filial generation, but later they separate, they react freely in F2 generation, second filial generation.